In this lesson, we're going to explore the structure and function of three different types of transmembrane receptors. And those three main types of transmembrane receptors that we're going to look at in turn are G-protein coupled receptors, receptor tyrosine kinases, and ligand-gated ion channels. Let's start with G-protein coupled receptors. These are involved in many processes, for example, several categories of sensory perception. Your vision, your sense of smell, and your sense of taste all depend on G-protein coupled receptors, and also the response to several hormones, like the hormone epinephrine. And they're a very important class of protein that are acted on by drugs. Many drugs have their actions by working on G-protein coupled receptor related pathways. So the steps involved in G-protein coupled receptor signaling are shown here in this diagram from Campbell biology. The ligand, shown here in red, binds to the G-protein coupled receptor, which is a transmembrane receptor embedded in the membrane, and that causes the molecule GTP, that's related to ATP, or adenosine triphosphate, but instead of adenosine we have guanosine. So GTP replaces GDP, which is the same thing but with one less phosphate on the G protein. So this G protein here of G-protein coupled receptor acts as a kind of shuttle, and it can hold either GTP in its activated state or GDP in its inactivated state. So the binding of the ligand to the G-protein coupled receptor causes GTP to replace GDP here on this G-protein shuttle. That G-protein shuttle then activates an enzyme called adenylyl cyclase, and the enzyme adenylyl cyclase converts ATP, adenosine triphosphate, to cyclic AMP, AMP being adenosine monophosphate. So cyclic AMP is abbreviated like this, small c AMP, and it's got a ring structure in it. That's why it's cyclic, and that's why this enzyme is called adenylyl cyclase. Cyclic AMP then is a second messenger. Remember that the first messenger is the ligand that stays outside of the cell, and the second messengers are the ones that carry the signal intracellularly. And cyclic AMP as a second messenger can diffuse widely through the cell, spreading the response. One of the things illustrated here that it is doing in this diagram is activating a protein called protein kinase A, which will then activate other proteins. So that's our first class of transmembrane receptors, the G-protein coupled receptors. Next are the receptor tyrosine kinases, and an example here is the response to the hormone insulin. When insulin binds to its receptor tyrosine kinase in the cell membrane, it ultimately results in the movement of glucose transporters into the cell membrane, and that's what allows glucose into the cell, which is one of insulin's main ultimate effects. Now, receptor tyrosine kinases are also often mutated in cancer, so they have important clinical applications. A little bit of terminology here. The word phosphorylation means adding a phosphate to something. I'll write P sub I for inorganic phosphate. So phosphorylation means adding a phosphate to a protein, for example. And adding and removing phosphates is a common way that proteins like enzymes are activated or deactivated. And a kinase is an enzyme that adds a phosphate group to something. There are other enzymes called phosphatases that remove them. Now, why does adding and removing phosphates activate or deactivate proteins? Well, phosphates are charged, and if you add them to a protein, then it makes that region more hydrophilic. That can change the shape of the protein, and remember that the shape of a protein dictates its function in large part. So adding and removing phosphates is a very common way that proteins are activated and deactivated. So a receptor tyrosine kinase is a receptor protein that is a kinase, so it adds phosphates to something. What does it add phosphates to? It adds phosphates to tyrosines, which are an amino acid. So that's what the name is telling us. The steps in signaling with receptor tyrosine kinases are shown here in this diagram from Campbell biology. Binding of the ligand to the receptor tyrosine kinase causes two of them to join together in a process called dimerization. A dimer is something with two similar parts. So here we're seeing dimerization as the ligand binds and then the two parts of the receptor tyrosine kinase come together. And then when it becomes a dimer, each part of this adds phosphates to the tyrosines shown here on the other part. That's sometimes called autophosphorylation. And then those phosphorylated tyrosines, again, tyrosine is a particular amino acid, those phosphorylated tyrosines can then activate other enzymes within the cell, and that's what you're seeing here, resulting in cellular responses. So that's our second class of transmembrane protein receptors, the receptor tyrosine kinases. Third up are ligand-gated ion channels. If this is a cell membrane where this is the outside and this is the inside, then in cross-section we might imagine a ligand-gated ion channel to look like this. If you look at it from the top, it might look more like a donut. It's got a core here of a channel that allows ions to pass through if it's open, and then this is the protein on the outside. So in cross-section it might look like this, and it can open and close, so I'm going to draw a little gate there. If there's a receptor part here, or part of the protein that is actually binding the ligand is over here, when the ligand binds, this gate may swing open, and that allows ions which couldn't normally travel through the membrane to move through. So it is gated or opened by a ligand, and it's an ion channel, hence the name ligand-gated ion channels. And a very common example of these are that they're in the membrane of the postsynaptic or the signal receiving neuron. So if this is a diagram of a synapse where we have one neuron sending a signal across a small gap here to the next neuron, the signal within the neuron is electrical, but when that electrical signal gets to the end of the neuron, it triggers exocytosis of vesicles containing neurotransmitter into the synapse, and those neurotransmitters act as ligands that open ion channels in this postsynaptic neuron. I've drawn it quite small, but you can imagine one of these binding to a receptor like this, opening an ion channel and letting, for example, sodium in. So the steps again are that binding of the ligand opens ion channels in the membrane, and that allows ions, sodium being one example, potassium will be another example that we'll see, calcium is another, to flow through the membrane. These wouldn't normally be able to flow through the membrane because they're charged and so can't get through the hydrophobic interior of the phospholipid bilayer. A specific example is that binding of the neurotransmitter acetylcholine, which gets abbreviated capital A, capital C, lowercase h, to the postsynaptic neuron, that's this neuron in this diagram, opens a sodium channel, allowing sodium to rush in and make the inside of that postsynaptic neuron more positive. And that actually constitutes the electrical signal that then will propagate down that neuron in turn. And we'll look at that process in more detail in the lessons on neural signaling. So that is a look at 3 different types of transmembrane receptors and their structure and function.